Hi, welcome back to my channel. Today our topic is red blood cells. Concepts that we are going to learn in this chapter, they have been asked throughout so many years and they are very important that we have to go in depth. We cannot skip on this at all. It's very important. So the concepts that we will be learning is packed cell volume, ESR, and osmotic fragility. These are very important concepts that helps in throughout the whole unit of blood. Now before we jump in depth of what they are, we can first jump into what a basic RBC is. Simple. RBCs, they are red blood cells. Very good. We already know that. So what's something new over here is that they are biconcave in shape. Well, that's again nothing new. 7 to 8 micrometer in diameter. The advantages of biconcavity. Why should they be biconcave? Well, because they can easily squeeze through the capillary walls. That is one of them. And when they are exposed to hypotonic environment, red cells withstand lysis to a greater degree, as well as they swell to a larger extent to attain a spherical shape before bursting. Let's say that I keep my RBC in a hypotonic environment, that is where it enables the RBC to swell up more. Because of its biconcavity, it enables it to swell to a good amount of size. It attains a spherical shape and still your RBCs do not burst. It can endure a huge amount of resistance before it bursts. Thus, they do not easily lyse when the blood becomes hypotonic. Hypotonic as in lesser concentration. So lesser concentration, that means more blood volume, like more watery fluid, due to which it tends to seep into the RBCs. RBCs become huge and spherical. Okay, now it also increases the surface area for the exchange of gases, that is oxygen and carbon dioxide. It's in the easy folding of the red cells and therefore facilitates their movement through the narrow capillaries. I already told this before that one of the major function for its biconcavity is the movement through its narrow capillaries. Now, basophilic stippling. This is like a type of an abnormality, which means that there are presence of numerous basophilic granules. They are dark, coarse blue granules that are present. Granules are like the small, not protein-like exactly, granule-like structures that are present. There are numerous of them, basophilic in nature. This condition is called as punctate or punctate basophilia. It is typically seen in lead and other heavy metal poisoning. It also occurs in thalassemia, megaloblastic anemia, infections, and liver disease. A very beautiful concept I actually want to share over here. Now, you see this word megaloblastic anemia. When you will read about the stages of erythropoiesis, you will notice that the RBC's original size will be around 20 to 25 micrometer. That is like a huge one, because the RBC's original diameter is only 7 to 8 micrometer. But then in case of basophilic stiffling, or where this would be seen in case of megaloblastic anemia, the RBC, while it is getting converted into a mature RBC, sometimes due to any defects or abnormalities, the RBC does not convert. It remains juvenile, due to which it is like a huge shape. That is why we call it as the megaloblastic anemia. Mega, very big shape. So megaloblastic anemia, infections, liver, lead, and other heavy metal poisoning. In such cases, you will see high amount of granules present in the RBCs. That is known as the basophilic stippling. Now, sometimes in lead poisoning, you will see a ring-like structure that is formed. That is known as the Cabot's ring, a ring-like, or a figure of 8. More likely, I would like to call the figure of 8 as infinity. But now, lipid bilayer. Lipid bilayer is common in any and every membrane. The lipid bilayer is primarily composed of phospholipid and cholesterol. I think you've already studied about this now. It has certain integral proteins, especially spectrin and chirin. Where it is? Yes. Band 3 protein, glycophorins, RHD protein. But the main ones are ankyrin and spectrin. Those are the two important proteins. Now, if something happens to the ankyrin and if something happens to the spectrin, it could lead to various type or different shapes of RBC will be present. The ankyrin proteins, they tend to form like the membrane skeleton. They're like the skeletal framework for the RBC membrane. Now the other proteins could be the other membrane or integral proteins, could be band 3 protein, RHD protein, and glycophorins. Band 3 proteins are like the anion exchanger, like it helps in the exchange of anions through the membrane. So you could understand that would be the function of the band 3 protein. So these and various ion channels along with band 3 protein. Now membrane skeleton, the two important proteins are ankyrin and spectrin. Now the common membrane defects, as I already told you, ankyrin and spectrin. So presence of ankyrin and spectrin in the membrane provides the biconcave shape, its deformability, so it is not easily going to just like break down or anything. Membrane stability and durability to the red cells. Well, ankyrin and spectrin, these are the star proteins of the RBC membrane. Therefore, deficiency of ankyrin or spectrin results in membrane defects like spherocytosis, ellipsocytosis, poikilocytosis, etc. What are all these? These are the different types of abnormally shaped RBCs. These defects also make the cells rigid so that the cells are destroyed prematurely. Now, the biconcave shape of the RBCs prevents it from bursting and easily breaking down. But when the shape of the RBC changes because of deficiency to ankyrin or spectrin due to various causes, the RBC membrane becomes so fragile and loses its stability such that it tends to break down very easily and they are destroyed prematurely. Okay, now there is something called as glucose 6-phosphatase deficiency, G6PD deficiency. G6PD deficiency causes hemolysis. Now what exactly happens? In one error of metabolism, red cells are susceptible to damage due to oxidation. Hydrogen peroxide, which is the oxidative damage that causes the damage, gets converted into water and oxygen. All right, presence of this G6PD, glucose 6-phosphate dehydrogenase. Because of this, it gets converted into this, and since this does not exist anymore, there is no damage to the RBC membrane, therefore preventing the oxidation of the RBC membrane. Therefore, in hemolytic anemia, that is anemia due to hemolytic disorders, there is a common deficiency of this enzyme G6PD. Therefore, especially hemolysis is facilitated. So what I want you to tell that in certain the function of the G6PD and in hemolytic anemia, G6PD is deficient. So you pretty much know what's going to happen to that RBC membrane. And also hemolysis is facilitated on exposure to anti-malarial drugs, though these individuals are generally malarial resistant. Now why are these individuals generally malarial resistant? It is because malaria depends on the life cycle of the RBC. Now because they have G6PD deficiency, that means oxidation of RBC membrane takes place. That means they have a weak RBC membrane, right? A malaria patient, let's say I have malaria. Okay, so what happens? Because I have malaria, I will have G6PD deficiency. Because I have G6PD deficiency, my RBC membrane is going to be very susceptible to damage. It will easily break down. And because the malarial mosquito depends on my RBC and my RBC is already weak because of the deficiency, I am naturally resistant to malaria. Works. So these individuals are generally malarial resistant. And hemolysis is further facilitated on exposure to the anti-malarial drugs. Now what are anti-malarial drugs? Now malaria as such, I am already oxidant. My RBC is already pretty weak. Now on infusion of the anti-malarial drugs, my RBC is not sufficient enough to take over that. So I'll explain that more in detail what I meant. Now people with G6PD deficiency are resistant to malaria because their red cells don't support the parasite's growth. But the same weakness makes their red cells break easily when exposed to oxidative drugs like antimalarials, because the antimalarial is an oxidative drug, but my RBC membrane is already under oxidative damage. Now on further infusion of oxidative drugs, that is the primaquinone, one of the most important antimalarial drug, that is primaquinone, and exposure to such oxidative anti-malarial drugs, my RBC membrane is going to further undergo more hemolysis. That means hemolysis is the breakdown of the RBC membrane. So that is what this concept is about. This concept will be throughout, that is going to be asked in biochemistry as well as in physiology. Packed cell volume. This is a very important one because it has been repeated so many times. Now think about it. Packed cell volume, something that is packed. Alright, so keep that in mind. It is also known as hematocrit. Hematocrit is the fractional volume of blood that the erythrocytes occupy on centrifugation. They accumulate at bottom as cells are heavier than the plasma. Simple. I take blood, I centrifuge it, plasma on the top, erythrocytes at the bottom. Because why? Because they're heavier than the plasma. And because of that, they tend to pack. Like the erythrocytes that are at the bottom, they tend to pack up. Significance of this packed cell volume. The packed cell volume is a reliable index of the red blood cell population. It provides us a useful information about the red cell mass, which is correlated with the red cell count in their hemoglobin content. Now, RBCs that are at the bottom are they're less in number. So you can correlate it with the red cell count and their hemoglobin content. So that is less. So accordingly, it is a reliable index. Now these measurements, that is PCV, the red cell count and the HB content, are essential for the determination of red cell indices that helps in determining and classifying the various types of anemias. Now what does this point mean? The packed cell volume, the hemoglobin content of the blood and the RBC content of the blood is very important in the calculation of the red cell indices. Now what are these red cell indices? By this I mean MCV, MCHC, etc. And there's even other one, MCH. Then there is something called as color index. Now these have certain formulas. Through these formulas, they have a normal range. Okay. Now because there could be variation in these factors, the normal range is definitely going to be either more or less. If on calculation of these values, your blood results in something more, you could say that you have certain types of anemia. Your blood picture is going to be like having macrocytic related anemia, and when it is less, it could be related to microcytic anemia, etc. So these are known as blood indices. This MCH, MCV, MCHC, color index, etc. These are the red blood cell indices that help in determine and classifying the various types of anemia. The various types of anemia, that is a whole new chapter, would be like iron deficiency anemia. Those are what the types of anemias. But according to this indices, this is what I just want to tell, and this is what you will be naming them. But the types of anemia are like iron deficiency anemia, vitamin B12 deficiency anemia, etc. It goes on like that. Okay. Then the normal value and variations in adult male, the PCV, the normal values and variations in adult male, it is 46%, in adult female it is 42%. You can take out the normal range, tell it 40 to 50, 37 to 47%. Now hematocrit, that is the packed cell volume, decreases in conditions of decreased red cell count, which we just said, decreased RBC, hemoglobin, PCV, they all indicate something. So hematocrit, that is PCV, decreases in conditions of decreased cell count and increases in conditions of increased cell count. Obviously, when the cells are deformed, as occurs in spirocytosis, mainly because of the defect in protein membranes such as ankyrin and spectrum. So when the cells are deformed, as occurs in spirocytosis or sickle cell disease, more plasma is trapped between the packed cells, which gives a false high result. Alright, so when patients have spirocytosis or sickle cell disease, you should just know that they're going to have a very high PCV because the plasma is going to be stuck between the erythrocytes that has been sedimented at the bottom. Now, ESR, erythrocyte sedimentation rate. It is also a very important one, has been asked many times throughout. And what is this ESR? The rate at which the red cells fall, that is sediment, is known as the erythrocyte sedimentation rate. Erythrocyte as in RBCs. Sedimentation rates, that means the red cells fall. Rate at which the red cells fall. So erythrocyte sedimentation rate. The important note difference between ESR and PCV, then what is that? What is the difference? Both of them are settling down to the bottom. Both of them are sedimenting. Well, over here in hematocrit, that is PCV, packing of red cells is accomplished by centrifugation. Remember? Whereas in ESR, the columns of red cells, they settle by gravity. In centrifugation, if you take a test tube and without centrifugation you leave, the erythrocytes will not settle down. But then over here in ESR, due to the gravity, the red blood cells will automatically settle down. So that is the difference between the ESR and PC. Factors affecting our ESR, effects of ESR, factors in the conditions, and how exactly is that happening. Now, factors. Why would our ESR increase? It would increase in case of increased fibrinogen. If increased fibrinogen is there, you can say that it is an indicative of infection in your blood. So because this fibrinogen, it promotes the Rolox formation. What is Rolox formation? It is nothing but like arrangement of RBCs in a form of coins, like coin-like arrangement of the RBCs on each other. This arrangement of the coin-like RBCs is known as Rolox formation. Increased fibrinogen increases the ESR because it promotes the Rolox formation by neutralizing the negative surface charge of the RBCs. Usually, the surface of the RBC will have a negative charge. Now, because of increased fibrinogen, it will neutralize the negative charge. It will bring it back to zero, causing them to aggregate and settle faster. This aggregation of the RBC is nothing but the Rolox formation. More the Rolox formation, more is the ESR, which is facilitated very well by the fibrinogen. What are the other cases it could increase? The other cases would increase in terms of increased globulins, especially the gamma globulins. So increased globulins, the mechanism is pretty much similar to fibrinogen. It is where they promote the aggregation as a whole. Okay, so then what are the factors where the ESR would decrease? Decreased ESR matlab decreased Rolox formation. Now decreased in case of increased albumin. Albumin resists the Rolox formation. It helps to maintain the negative charge. The negative charge is also known as the zeta potential. The negative charge in the RBC membrane, that is known as the zeta potential of the RBC membrane, it is very well maintained by the albumin. Now, when the albumin increases, it increases the negative charge or the zeta potential of the RBC membrane, causing them to repel each other and settle even slow. Again, it decreases because of high blood viscosity. High blood viscosity, hey there, we'll be talking about fibrinogen also. Think about that. So high blood viscosity physically impedes the fall of the aggregates, slowing the ESR. Now, other reasons it would increase is because of the presence of macrocytes. Now, the macrocytes that are there, that are nothing but the large RBCs. Now they settle faster due to the smaller surface to volume. This is according to the Stokes law. So the Stokes law, you could pretty much understand that like more the surface volume ratio, like the larger the size, smaller the surface volume ratio related stuff. So they settle faster due to the smaller surface to volume ratio. Now certain conditions where the ESR would further decrease besides increased albumin and increased viscosity would be polycythemia, that is high RBC count or high packed cell volume. Now increased blood viscosity and greater inhibition of Rolox formation is due to the higher volume of the RBCs. Now decrease in microcytes, that is the small RBCs, where they settle slower. Now abnormally shaped RBCs like spirocytosis, poikilocytosis, ellipsocytosis, in all of them, or the sickle cells that you will see in sickle cell anemia, their irregular shape impedes the Rolox formation, preventing them from stacking and setting quickly. So you can understand that the patients who will approach to you with sickle cell anemia, you can keep that in your head. They're going to have a decreased ESR because of their irregular shape preventing the Rolox formation of the RBCs. Now next, again, increase. Advancing age. This is like a physiological condition. With increased age, more is the Rolox formation. With the female sex will have it's slightly higher than males mainly due to the presence of the hormone estrogen, and increases in case of pregnancy after the first trimester due to the increased fibrinogen content of the blood. Now, the causes for increased ESR again. So let's overall, until now you saw the factors and the conditions. Now what could be the causes for this increased ESR? Now the presence of inflammation, it was related to fibrinogen. So inflammation that you could see in acute phase reactions. So presence of inflammation or infections also due to an elevated level of acute phase proteins, especially fibrinogen in blood, which promotes the Rolox formation. How? It removes the zeta potential of the RBCs. So therefore, inflammatory or autoimmune diseases, infections, malignancies such as cancer, which is very dangerous. So you can say that, hey, you have an increased ESR because your fibrinogen is low. That is because you have an inflammation or acute phase protein something, or you have a malignancy, multiple myeloma. Multiple myeloma, it is where your AG ratio is going to be reversed. You know, the normal AG ratio is 1.2 or 1.2:1 or 2.2:1. But over here, the globulins will usually be at a higher level than albumins. In multiple myeloma, there is decreased amount of albumin. Therefore, it does not help in the negative charge of the zeta potential of the RBC membrane, and therefore, in cases of multiple myeloma where the AG ratio is reversed, that is albumin decrease, globulin is going to increase, which is unusual, it would cause an increased ESR. Now, causes for the decreased ESR. A low ESR indicates that the red blood cells are settling down slower than the normal. Now, the RBC or the blood disorders are like polycythemia, that is increased RBC count, decreased number of RBCs, etc. They all tend to settle down slower. Sickle cell disease or hereditary spherocytosis, like abnormal spherocytosis. The plasma protein conditions, hypofibrinogenemia, that is low fibrinogen. Fibrinogen, it tends to neutralize the charge, increases the Rolox formation. But now if there is decreased fibrinogen, the negative charge does not neutralize. Therefore, lesser the Rolox formation, there will be the decreased ESR. Now, the normal values of the ESR, the Rolox formation in your blood is normal. But what is that normal range? There are two methods according to which you will perform the ESR. There are two methods, Westergen method and the Winthrop method. So the normal values vary for each one of them, but the varies is not that much. Yes, ESR is obviously going to be different between males and females. In Winthrop method, in males, it is 0 to 9 mm per hour. In females, it is 0 to 20 mm per hour. But in Westergen's method, in males it is 3 to 5 and females it is 5 to 12. It is a physiological reason as to why females have increased ESR than the males, mostly related to the hormone estrogen. Now, this Rolox formation description, this is what I was mentioning, the coin-like stacked arrangement on each other. So they resemble stack of coins. Now it is a condition, this is a picture where I referred it from GK Pal. Now this is seen when the gamma globulin is increased and ESR is high. Multiple myeloma, Kala Azar, chronic inflammatory diseases, cancers, etc. This is where you'll see the variations in the ESR. Now, osmotic fragility, that is our last topic, and this also has been asked so many times throughout the year. So osmotic fragility, fragile. So osmotic fragility is defined as the ease with which the red cells are ruptured when they are exposed to the hypertonic solution. Now, before we jump into the concept, according to whatever we've learned right now, let's just try. You know that the RBCs are able to burst easily when they are exposed to a hypertonic solution. That means something must be wrong with the RBC membrane, something must be wrong with ankyrin and spectrin. So their hypertonic solutions, after, in spite of attaining a spherical shape from biconcave, it is still bursting. So that shows the osmotic fragility, the ease with which the red cells are ruptured. It assesses the integrity of the red cell membrane. The osmotic fragility test helps in the diagnosis of anemia in which the physical properties of the red cells are altered. Anemia like megaloblastic, microcytic, iron deficiency, all of the anemias will have different blood pictures with different shapes and different protein deficiency. Therefore, anemia could be one of the causes where you would see osmotic fragility in the RBCs. Now, in an isotonic solution, where such a solution has the same tonicity with that of the plasma, examples are 0.9% NaCl. This is the main one. The rest, if you want, you can memorize it or not. But we will be taking the example of 0.9% NaCl throughout. So this is where the solution has the same tonicity. But when the RBCs are suspended in a hypertonic solution, the water or like the RBCs tend to shrink. So when suspended in a hypertonic solution, a solution with more tonicity, that means more concentration of the solute than the solvent, the red cells shrink due to loss of water from them by exosmosis. The opposite happens in terms of that is hypertonic solution. The red cells absorb water by endosmosis when kept in hypertonic solution. A solution with less tonicity, that is less than 0.9% NaCl. Endosmosis results in hemolysis due to swelling and rupture of the RBCs, because RBCs, they take up the water, attain spherical shape, and then eventually burst out. That is known as hemolysis, breakdown of RBCs. Now, increased osmotic fragility denotes the decreased ability or the decreased resistance of the cells to rupture. Usually the RBC cells will resist pretty much, but if there is decreased resistance, they're easily going to burst open. So increased osmotic fragility, decreased resistance of the cells to rupture, which is related to its geometric configuration. So if the cell is biconcave, it will have a good resistance. But if it is not biconcave, it could be any other shape, abnormal shape, it would cause decreased resistance and therefore increased osmotic fragility. The red cells that are spherical demonstrate increased hemolysis. That means increased osmotic fragility, whereas the red cells that are flat like sickle cells demonstrate decreased hemolysis. So you should know that spherocytosis increased osmotic fragility. Sickle cell shaped will have decreased osmotic fragility because they have more resistance and they demonstrate decreased hemolysis. Now this is how the picture that would look. I was talking all this time in packed cell volume, RBC count, like hemolysis, whatever I was talking, the shapes of RBCs. You could see this is how a spirocyte would look like. This is how a normal RBC would look like. Spirocyte, the blood vessels. So normal macrocyte, microcyte, ellipsocyte. Look how beautifully each one of them have been represented. The teardrop cells, the values, and the decreased fragility. Where do you see the decreased fragility? But what are the normal values for fragility? The normal value and variations. Normally, the osmotic fragility begins at around 0.45 to 0.50 and it completes at 0.30 to 0.33. That means it starts to resist the osmotic fragility. Let's say it is around 0.45 to this, I can still resist, but once it goes to 0.3 to 0.33, it's going to become extremely osmotically fragile. That means I can easily break. Now, conditions of diminished fragility: iron deficiency anemia, thalassemia, sickle cell anemia, obstructive jaundice. Diminished osmotic fragility. What does this mean? Diminished osmotic fragility, as in there is more resistance of the RBC due to which they are less osmotically fragile. That we already know that we'll see that in sickle cell anemia. Now you're going to know that it is in iron deficiency anemia, thalassemia, obstructive jaundice, and post-splenectomy. Now the conditions where you would see increased fragility: spirocytosis. So hereditary spirocytosis, congenital hemolytic anemia are the conditions in which the spirocytes are found in the blood. That's it. Simple and easy. This chapter is actually pretty important. Keep on listening to this video again and again if you haven't understood this pretty much. I really hope I reached out to you and I made myself very clear about the concepts to you. I don't feel pretty deep fried because I felt the chapter was pretty good, but if you did, it's okay, chill. That was pretty much my condition when I finished this chapter when I was in first year too. I was dead fried. So definitely do like, share, subscribe. Alright then. Have a nice day. Bye bye.